Anything else specific?
Can we go over natural versus artificial immunity and then how that would connect with active and passive? So like maybe an example from each category.
Yeah. Let's see. And I actually looked this up in the book, Sarah. I think it's chapter 9 instead of whatever chapter it says that it is on the PowerPoint. Let's see.
Yeah, the chapters don't line up with the PowerPoints. But I found that looking in the homeworks, it'll have like multiple choice chapter number versus problem number. So you
Oh,
can actually look it up that way too.
nice. That is awesome. Let's see. Slide. Okay, cool. So yeah, active, you or the host or person is getting infected or exposed somehow to the virus, and they are developing an immune response in their body. And then passive immunity is like the really good example of natural passive. Passive is breastfeeding when mom is giving antibodies. And then what was it like? Is it IgG, IgA, IgG? That's passed through breast milk. Yeah, and then artificial passive is, I think of, okay, the vaccine is artificially made and like it's already developed, it's already antibodies against the antigen, so the person is not making the antibodies against it, right? And so it's getting put into the human, but the immune response already happened in another organism, like a mouse or an animal. And then artificial active would be like the vaccines we talk about, like the attenuated or inactivated vaccine. So artificial passive immunity would be like COVID antibodies, that are taken from a patient that has already recovered from COVID.
Nice.
Yeah.
Thank you.
Absolutely. And the natural active COVID parties or chickenpox parties. You hear about people doing that crazy stuff.
I feel like that's going to be one of the essay questions for sure.
Yeah.
I just had a real quick question. So is inactivated for like the vaccines, is that artificial active?
Artificial active. I would say so, yeah. Yeah. Because you're, let's see. Can you guys still see my screen? Because they can induce an immune response, but then you give like the adjuvant to stimulate the immune response in the person that's getting the vaccine. So yeah, I think that would be artificial active, right? Yeah. And I think this was a question too on the document, to know the difference between attenuated and inactivated and the advantages and disadvantages. That would also be a good like short answer question. And maybe examples of both. So here are some examples of inactivated.
When he discussed the direct and indirect like ways to label antibodies or antigens, I guess I don't really understand like the advantage of one over the other. Could you explain that a little bit? Or even the like two different methods of ELISA, if that one's easier? Just why you would do one over the other?
Right. From currents. let's see, direct versus indirect. I don't think there's any, you know, advantage or disadvantage to either of these. The indirect, you just have an extra step, an antibody that binds to the antibody. So, I don't know if that helps at all. But
If you don't feel like it's going to be very important, then that's fine. But I was just trying to kind of like separate them in my mind a little bit.
okay, well, direct, you're definitely interacting like right with the antigen itself, and then indirect, the antibody that's going to be the visual result is binding to the antibody that binds to the antigen in the like indirect way. But for a direct one, the antibody that, fluoresces or gives like some kind of visual label is binding directly to the antigen. Does that help?
Yeah, it does. Thank you.
Cool. But yeah, I don't know if there's any advantage or disadvantage.
Do you feel like we need to know the details about them too?
Um,
Like every detail, explain from like here to the end, and then for the stuff like that.
maybe for the ELISA, I could see him like, this is just kind of an example of, in general, a direct test binds right to the antigen and an indirect binds to the antibody that binds to the antigen. They call that the secondary antibody. But I could see him wanting you to know how an ELISA works, right? So this was just kind of like the background information for the direct and indirect ELISA. Let's see. And so basically the way this works enzyme linked is that the antibody that binds to the antigen has an enzyme that cleaves some kind of substrate, and when it cleaves that substrate, it gives a visual color. And so that's like the pregnancy test is an example of an ELISA with the colored lines.
Do you think that might be a question that he would want us to give some examples of ELISA tests?
Maybe. And also, I believe that that a pregnancy test would be a direct ELISA because it would be trying to detect hormones directly in the urine. So I think that would be an example of a direct.
What's an example of an indirect?
That's what I was trying to think of. Let's see. I feel like maybe an HIV ELISA is an indirect. Let me Google it though.
We also did the one in genetics for the plasmid.
Yeah, we did a simulated HIV outbreak.
Yes. Yeah, that's what it was. The PGLO is to show how the plasmid DNA
Yeah.
went into the bacteria cells.
Yeah. I think that's on our next micro test, possibly. Let's see. Honestly, you could probably do either type of ELISA for different antigens. It's just, are you using the labeled antibody to bind to the antigen, or are you labeling the secondary antibody? It just differs in which one's the visual cue, or which one's the visual antibody. Let's see.
Did Dr. Daft ever post those slides he said that we should look over from today?
Ooh, the extra coronavirus slides.
Yeah.
Let me text him. He said he was going to change the PowerPoint, but I opened it, the virus one.
Is it anything he didn't go over in class, or is it just stuff he added last minute and went over in class?
Just stuff he added about coronavirus. Let's see. Here's the virus PowerPoint. Okay, I don't know if I'm seeing anything on COVID. Yeah, I can shoot him a message really fast and see if he was gonna upload that, because he did say he would. And I just Googled, and it said that you can use like a secondary ELISA to find specific antibodies in a serum. Like if you have a sample of a ton of different antibodies, then you can use a secondary and indirect ELISA to find a specific antibody that you might be interested in. So maybe an example of that would be like the COVID antibody test. Let's see.
And that would be a direct, or which one for the COVID?
I think that would be indirect. Like if you want
Indirect. Okay.
yeah.
All right.
But a direct example would be exposing it to COVID antigen. So you could do either. Does that make sense?
No, actually, you said indirect would be COVID-19 test.
Well, the COVID-19 test, if you could probably do it either way, right? So if you want to directly see if the antigen is there, then that would be a direct ELISA, or the antibodies. So you could expose like a sample to the COVID antigen, and if there's a positive result, then that would be a direct test. But an indirect test would say, oh, I have this sample of serum that might have antibodies in it, and if you happen to have an antibody that binds to the COVID antibody, that would be an indirect. Does that make sense?
Perfect. Got it. Yep.
Yeah. Okay, let me text Daft about the PowerPoint. Cool. I could see, I don't know if he would ask this, but I could see like a question about types of like ways to do immunoassays. But I don't know. Maybe it won't be that specific.
Like an essay question, you think?
Principles of immunoassays, or maybe multiple choice.
That one. Hey, can Lisa? Can you explain? Can you explain? No. Keep going back. That one right there.
This one?
I do not understand. Yes. Yeah.
Let's see. Let me do this from the beginning. It's not what I wanted to do. Sorry, guys. Oh, here we go. So let's see. Unknown organism plus a solution with known antibodies. Okay. So I think this is just saying if you have an unknown organism, but then you have antibodies that you know bind to it, or know that you bind to a specific pathogen, but you don't know what this organism is, but you know what the antibodies bind to, and you put them together, and there's a reaction, so the antibody binds, then you know, okay, this unknown organism is this specific pathogen, the Treponema pallidum. That's probably not right, but and then if you know the patient's like you don't know what their antibodies bind to, but you know what the pathogen is, you can see, oh, is this patient immune? Has this patient been exposed to this specific pathogen? So it's just kind of like a similar principle, but just like you might know one piece of information or the other. Does that help, Holmes?
Got you. Yes.
Yeah. So that would be like a COVID antibody test, right? Like you could do this either way. Antibodies of a patient, you take it out and then you expose it to a COVID antigen, and that would be a test to see if that patient's been exposed to COVID, or if they have antibodies that have been made against a COVID antigen, which would suggest that they had been infected previously.
All right. I think that's it for me. Thank you.
You're welcome, home. I think you guys are gonna do fine. You made it through the first one.
So if I'm a little confused because there's 5 things that antibiotics can target, but 4 mechanisms that they can use to become resistant.
Right.
So why is that?
Like, why is it not like a one-to-one thing?
Yeah.
Let's see. I don't know if you guys. I stopped sharing my screen. It's probably that one mechanism just can affect multiple things. Let's see. So here's the mechanisms: inhibit cell wall protein synthesis. Okay, I bet what's related is like the membrane and cell wall stuff. But let's see. Mechanisms. Oh, okay. So I think this is just like for mechanistic categories, like it could be resistant to the medication, right? Like somehow, oh, let me make this slide bigger so we can talk about it. Somehow inactivate the enzyme like the beta lactamases, or increase, like upregulate efflux pumps to pump out the drug, or somehow alter
Oh,
the.
it's not sharing the larger one.
Really?
I think, yeah, I think it makes you share your screen.
What the heck? Let me see. Let me just try to make it bigger then. Is that better?
Yes, that's better.
Okay, cool. And then let's see. Another way to acquire resistance is through like plasmids that might have antibiotic resistance or something like that, right? Or just random mutation that then gets passed on to daughter cells. So that, yeah, that's mutation and horizontal gene transfer. But I also think that would be a good short answer, is like how do pathogens become antibiotic resistant, or sorry, how do bacteria become antibiotic resistant, or medication resistant? That's good. So yeah, being able to describe, okay, the bacteria might upregulate efflux pumps, and then you're pumping out the drug and it's not having an effect. Or you express an enzyme that cleaves the antibiotic and it doesn't have an effect. Like being able to describe these different things about the picture. That's another way I would study too, Lauren, is like if you have a figure in the book, like read the paragraph about it and take notes, and that's really good ways to beef up your like short answers.
Okay, yeah, because it's always like those types of things that get me, because I feel like there's so many little moving parts. So that's a good idea. Thank you.
Yeah. Because I know it's hard to like read every little thing in the book, so that's a good place to start.
So when it modifies the antibiotic, is that similar to it binding to the wrong target? Like looking at the top picture?
Let's see, antibody target. Antibody binds target. Okay, so that's in a non-resistant cell and what normally happens. So what could happen, let's see, alteration in target molecule. So it could modify the target in some way, like chemically alter it to where it doesn't recognize the target anymore. So the bacteria could, slightly mutate the binding site for the antibiotic, right?
Okay. Then with the antibiotic inactivating enzyme binding to it, would that have to like copy the shape of the binding site?
Enzyme modifies antibiotic inactivate. If it was going to bind to the new target?
No, if the enzyme was going to bind to the antibiotic, would it have to mimic the target shape in order for it to bind properly?
Oh,
Or would
yeah.
it bind somewhere else?
No, you're right. It would mimic.
Okay.
It would have a site that mimics the shape. Yeah. For sure. Oh, there is a chat. Sweet. Yeah, Abby said, hey, Alyssa, she's feeling good about it. So she peaced out. But yeah, this stuff is kind of like Abby said earlier, it's kind of more intuitive. I think. Oh, and Sarah, just because I'm an evolutionary, that makes me think of if the bacteria would, somehow find a way to mutate the target molecule, then only the bacteria that have that certain mutation are going to survive. So there's just like the selective pressure. Yeah, just random chance that it would have a mutation in the target molecule.
Yeah, I know we talked about antibiotic resistance a lot. Do you think that'll probably be an essay question or a short answer question?
I think so. And like the mechanisms of resistance probably, and like the implications of antibiotic resistance. We talked a lot about that, like from a, healthcare professional perspective. Yeah. And I think there's the TED Talks on Moodle. Like I think those would
Yes.
be good like ways to pull questions.
Yeah, I noticed when I was watching the TED Talks that it didn't give the direct YouTube link. It gave a link of like screen sharing over it, and there was questions typed onto the image of the video at certain parts of it. So I need to go back through and rewatch those.
Yes, those are good to know. And I'm glad we're doing this on Wednesday and not tomorrow.
Yeah, it gives me time to get a list of stuff to do for tomorrow.
I'm so glad. My roommates are playing Mario Kart.
That sounds like a lot of fun. So the questions from the research papers,
Yeah.
should we go through the document that has like research paper questions in it? Or do you think it will be similar to the stuff that was on the quiz?
I would definitely go
Because-
back and review the quizzes, and then those questions, that's what he did for us. He had us read the article, and then we had to take quizzes over the articles that were written with those questions. And so those were what he gave us, and those were similar to what were on our tests. So I would definitely try to go back and answer them just to be safe.
Okay, because the questions on the list that were on Moodle are more in depth than
Like those require that you have like read the whole thing. On your last test, were there any questions, like essay questions about the articles?
No, it was mostly multiple choice questions.
About the articles? Cool.
Mhm. Yeah, the multiple choice questions were mostly class stuff. Give me a second, I can pull it up. There was a short answer about the IgM versus IgG levels from one of the COVID papers.
Yeah, like active or previous immunity. Yeah.
Yeah.
So yeah, I think, and like what he
Okay.
touched on in class in our article discussion, like that would probably be good to know. Fecal microbial transfers. But yeah, those questions that say like the research questions, the ones that are really in depth, like those would be good practice for like an essay question.
Yeah, I felt like most of the essay questions were stuff that we went over a lot in class.
Yeah, that you could tell were obviously going to be an essay question.
There's one that was an essay question that I wasn't really expecting to be. Well, I guess it was a short answer. It was worth 5 points. But like the epidemic versus pandemic versus endemic.
Oh yeah. Honestly, micro it's so much like compare and contrast, like different things. So any of those types of things are going to be great, like great short answers.
Yeah, and then the long answer essay question was, you've been infected with a bacteria, what happens next?
Oh wow.
And so giving the whole innate and adaptive connected response.
Yeah.
I have a question.
Yeah.
It's probably dumb, but it was like kind of what we went over in class today. So the bacteriophages, so is that a virus attacking bacteria?
Yes. With the capsid and the little feet spikes.
Yeah. Yeah, so it is a virus. All right.
Yeah, phage is a word for virus, and so you're right. Bacteriophage is like a virus infecting bacteria.
All right. I think I might have missed that. And so like the whole part of the lecture he was talking about that, I was super confused because I was like, so is it bacteria affecting bacteria? Yeah, that makes more sense. I didn't want to ask it in class either because it sounded dumb.
Or you could just type it in the chat.
Yeah.
Or you could say bacteria infecting virus, I guess that would be the way to say it. Virus that infects bacteria.
All right. All right, thank you.
You're welcome, McCann. You guys got anything else?
No, I think I'm gonna split.
Okay. Yeah, you're gonna do great, Michaela. Oh, thanks, Michaela. You're gonna do great.
All right, I think I'm good too. Thank you.
You are welcome. I'm always like hoping this helps.
It does.
Do it like from now on. We'll do it like a couple days before because I think this was better.
Yeah, and it gives more time to study because last time this was the last thing I did before the exam.
Yeah, so it feels a little pressured.
Yeah, and I don't like staying up all night trying to study because I know I don't do as well when I haven't slept enough. So
Right. This is true. All right, Sarah. I'll
All
see you tomorrow.
See you tomorrow.
Bye.
Bye.